Right. So in this part, you are going to test if glucose actually is glucose produced by the process of photosynthesis. That is this activity. And the next activity, 11.2, is oxygen really necessary for this photosynthesis? Okay, so what are the questions, Puta, over here that must be in your head? The first activity targets. Sarah, listen to me very carefully. The first activity targets. The first activity targets is glucose actually produced during this photosynthesis. First activity. The second activity is oxygen really produced during this process. Make sense, Puta? Right.
Yes, miss.
Now, even though I keep on telling is glucose being produced, is that is the experiment, even though I keep telling, they have not used the word glucose, no? They have used the activity to test if starch is produced. Why is that, Puta? Listen to me very carefully. That is because, let me make this one page so that it's clear. Right. Here, see, Puta, they have not used if the leaf contains glucose. එහෙම කියලා මොකුත් නෑ මෙතන. If the leaf contains starch. You know why, Puta? Listen to me very carefully. When photosynthesis take place, in one place, Puta, now what is being produced? Glucose, no. But the leaf won't keep it as glucose. The leaf will convert the glucose into starch and only keep. Does that make sense? There's a
Yeah.
conversion that takes place. So you can't test for glucose. What you have to do, if you test for starch and if starch is present, that means glucose has been uh, uh,
So do we have to change that formula thingy?
no, Puta, no. This is photosynthesis. The equation that they gave you, this is only photosynthesis. But your plant, the leaf is doing an additional step. Here, see, glucose produced
Ah,
by
so
the
it
leaf
just
is
transfers the glucose into stuff.
into starch. It's an additional step, Puta. So, what we do, we are going to test during photosynthesis is glucose being produced through the testing of starch. If starch is present, then obviously glucose was also present. Make sense? Okay,
Yes, miss.
right, Puta. Okay, so the very first experiment, let me explain it to you all. So can you all see here? Huda, I hope you also can hear me clearly. Now this setup is very easy, Puta. Listen to me very carefully. I will explain it to you in a very simple way. What is the purpose of this experiment? You are going to check if starch is present, okay? In order for you to check for starch irikida illaya and pakrathukku, if starch is present or not, the simple experiment is we call it as the iodine test. Iodine test, right? What will happen is iodine is put a brown in color, brownish yellow color. Harida, if you have starch on this one, let me draw the starch in maybe pink color. Oh no, not pink color. Uh, Green color starch. If starch is present in this sample, if I put a drop of iodine, if I put a drop of iodine, the iodine color will quickly change into purple color, purplish blue color. Okay, or we can tell dark blue. So what is the starch test, Puta? I'll tell it to you simply. Starch irikida illiyana paakarathukku, what do? We do the iodine test. How is the color change will take place? If starch is present, if starch is present, what would happen, Puta? Brown color will turn into, brown color iodine will turn into what color? Dark blue. Easy.
Yeah.
Understood? Right.
Yeah.
Now, it's a very simple test now, right? All you have to do is take a leaf, put iodine and see for the color change. But why are we doing all these steps? That's because, Puta, the leaf is green in color now. The leaf is green in color. So can you see the color change clearly when the background is green? No. So we add a step. So we add a step to remove the greenish color of the uh, leaf. කොහොමද අපි ඒක කරන්නේ පුතා? We are actually doing it by boiling the leaf in alcohol. We are going to do this double boil thing. Okay, in boiling in a water bath. Right? බලන්න මෙතන ඉස්සෙල්ලාම you are going to boil the, here see, look at this one. Uh, the figure 11.2, boiling in water. Dip the leaf in hot water and boil. Put them into a boiling tube with alcohol, and then boil it again. So you're going to boil it twice. Number one, number two, right? Okay. Does that make sense?
Yes, Miss.
Right. Okay. All right. Now, if I were to go down, you will observe that the leaf turns into dark blue color when iodine is added. Iodine is an indicator. So what I told you over here. Now, then, what do we tell? To remove off the greenish nature of the greening pigment of the uh, leaf only, we are going to boil it in the alcohol. Okay, right, until the leaf becomes colorless. Can you see? The leaf will become colorless. Okay. Now, Puta, I want you all to grade 8 to write the heading. Please write the heading. Test whether glue photosynthesis, uh, test whether photosynthesis, during the process of photosynthesis, if starch is produced or not. Okay. Write the heading. All right. Write the heading and draw this structure, Puta, soon. Okay.